So Robin, we're recording. Okay, well, welcome everybody, and welcome to another Purdue Lecture Hall series. My name is Tommy Soares. I'm the Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And today I am so honored to welcome Dr. Caroline Plescia, who just passed her PhD defense. And congratulations, Dr. Plescia. I am sure you're feeling quite accomplished and quite relieved at the same time. Quite a feat to have gone through, so congratulations. And we're really excited to hear you talk today a little bit about your work and what your adventure has been so far in the area of research of these very dangerous viruses that you study, like Ebola and SARS CoV-2, and that you're actually building tools to make them not dangerous to study in the lab. I think everybody on the call today and watching this video today is going to really get a great lesson as to how we do this. And I'm really excited to get us started. So Caroline, without any further ado, please take it away.
Thank you. Okay, so as Tommy said, I'm Caroline Plescia. I'm going to be talking to you about the work I've done at Purdue over the last 5 years, and that is designing molecular medicine to prevent virus infections. So these are a whole bunch of different viruses which infect humans and animals. And you can see that each virus looks a little bit different, but some of them look similar to each other in that some of them are circular and others have more odd shapes, such as the Ebola virus. So these structures are made up of individual proteins that the virus encodes. And you can see that viruses that have similar structures have similar proteins, but viruses that look different have really special proteins that no other virus has. So I'm going to be talking today about specifically Ebola virus and its proteins, and also the coronavirus and its proteins. But first, we're going to start with Ebola. So in sub-Saharan Africa, there are often outbreaks of Ebola virus, which causes a severe hemorrhagic fever in humans, which is a super serious disease that causes you to bleed out. So you don't want to catch Ebola virus. So it's really dangerous when this virus breaks out. And it was specifically discovered in the Democratic Republic of the Congo, alongside the Ebola River, which is where the virus gets its name from. So like I showed you earlier, the Ebola virus has a special structure, which looks like this. It kind of looks like a shoestring or a long filament. And this structure is made up of 7 specific proteins, which are all labeled in this image. But we're going to focus on 2 special proteins of the Ebola virus, which give it its structure. And that is the matrix protein, VP40, which is this brown protein right here. You can see it on the inside of the entire virus. And then also the glycoprotein, which is this yellow protein that sticks through the envelope and helps mediate viral entry. So usually in order to study this virus, you need what's called a BSL-4 lab. And that's a big special lab where you have to wear spacesuits inside of it and be hooked up to oxygen. And that is because you're working with really dangerous pathogens. And there are only about 13 of these quote unquote BSL-4 labs in the US. And the scientists that work in those labs just don't have the time, money, or resources to do all of the experiments that need to be done for these pathogens. So that's why we look at models that we can use for Ebola virus that are either BSL-3 or BSL-2 compatible. And that's because there are exponentially more BSL-3 and BSL-2 labs in the US. There are hundreds of BSL-2 labs on every major college campus, so you can find them pretty easily. And so we wanted BSL-2 models of Ebola virus called virus-like particles, which I'm going to call VLPs. I'm going to talk a little bit more about what VLPs are in a subsequent slide. Just know that you can use them in a regular lab to study viruses that you couldn't usually study in a regular lab. So this is what happens when the Ebola virus infects a cell. So this is a big overwhelming chart. So let me break it down for you. This is the virus, the Ebola virus, and over here is a cell. So right here, this gray line is the plasma membrane, which separates the extracellular space from the cytoplasm. So the virus will come and interact with this plasma membrane, and it will cause these ruffles to form, and these ruffles will engulf the virus and package it into a little vesicle. Inside of this vesicle, the virus traffics deep inside of the cell, and eventually there's a pH-dependent fusion mechanism which releases the genome into the cytoplasm, and this is when an infection is established. So once inside the cytoplasm, this genome can be replicated, and it can be used to produce viral proteins. So all of the viral proteins will go off and do their individual viral functions. Each one does something special. And one of them is to replicate the genome. So once the genome is replicated enough, it will be taken to the plasma membrane, where it will bud out into the extracellular space and make a new progeny virus, which can then go on and do the whole life cycle again with the same cell or a different cell. So my project starts focusing on this drug on the left, which is called Diphylline, and a group of phenol-based derivatives of this drug. So basically we took this group right here and we cut it off and we added different atoms to see if we can make a better Diphylline molecule. So those ones are called synthetic derivatives of Diphylline, and I'll show them in just a minute. And Diphylline is a natural product, not our synthetic derivatives, but the original molecule. And it is isolated from this tree, which grows in China and is called the happy tree. So we wondered whether or not Diphylline and our synthetic derivatives would inhibit Ebola virus infection. And that's because Diphylline is known to do something special to the cell, which we thought might inhibit the virus. So on the left, what you're seeing is the raw data that our BSL-4 collaborators gave us from the virus studies. So what you're seeing is on the bottom, increasing concentrations of drug, and on the side, you're seeing how much of the virus is alive. You can see at the lowest drug concentration, 100% of the virus is still alive. But as you increase the drug concentration, moving to your right, you can see that the virus starts to die. And so at our highest drug concentration, almost 0% of the virus is still alive and functioning. So this means that all of our drugs, the Diphylline and our synthetic derivatives, do inhibit the virus. So we were super excited by this, but this was the last thing that our BSL-4 lab could do for us. So we needed to use those BSL-2 models in order to study the virus. So what we did was we wanted to look at the cell and specifically the uptake of viral particles into the cell. So what I'm showing you is the ruffling of the plasma membrane and how that can package into a vesicle and traffic it into the cell. And so what we did was we studied this pathway, but we introduced a virus particle. It gets taken up into these little vesicles, packaged, and then trafficked deep into the cell. And at some point, it will leave the vesicle and escape into the cytoplasm. So like I said earlier, the way that we were able to study this virus in a BSL-2 lab was using virus-like particles, which I'm going to call VLPs. VLPs look a lot like the authentic Ebola virus, but they lack the viral genome. So they're just kind of hollow on the inside. And we make these VLPs by expressing only 2 to 3 of the structural proteins, which give rise to the virus structure. But they don't have the rest of the proteins or the genome, so they can't replicate inside of cells. So again, going back to our virus life cycle, we can model all of these steps with VLPs except for this one because there's no genome. But if you use plasmids, you can make VLPs at the plasma membrane. You can then collect them and use them to model viral entry. So I just mentioned the word plasmid, which I'm sure many of you are not familiar with. But a plasmid is a little piece of DNA that you can encode whatever protein you want in, and then you can give it to cells and they will take up that DNA and express it. And that means they'll produce whatever protein you encoded into that DNA. So this is something we do every day in the lab. we grow our little cells in a little plate that looks like this. You can look at them under the microscope and you'll just see a whole bunch of irregularly shaped cells next to each other. And this is what it looks like. So all of those dark rough patches are cells. So I took my cells that looked like this, and then I gave them my plasmid, which encoded for a green fluorescent protein called GFP. And that is just a protein that glows under the microscope. So you're able to actually see it. This causes our cells to turn green, and you can see them under the microscope like this. So this is how we essentially put DNA inside of cells, and they can express whatever protein we want. So if you exchange plasmids for that GFP and encode the Ebola proteins, VP40 and glycoprotein, the cell will take up those DNAs and express those proteins. And what you start to see is the formation of Ebola virus particles at the plasma membrane. So right here is the inside of a cell. This dark spot or this dark line is the plasma membrane, and all of these dark filaments that are budding out of the plasma membrane are Ebola virus particles, and they're budding into the extracellular space where the cell will release them, and then they'll just float around until they find another cell to infect. So if you take the media off of the cells where all of these viral particles are floating around, you can centrifuge them on top of a sugar cushion. So that's just basically a few milliliters of sugar. And that will cause your VLPs to pellet. And so at the bottom of the centrifuge tube, you get a big white pellet, and that will be your virus particles. And this is what a purified virus particle looks like. You just get a nice shoestring-like structure, and that is just made of the two structural proteins, VP40 and glycoprotein. So we can do the same thing, but this time I'm going to put a GFP specifically on that VP40 protein, and it will go to the plasma membrane like it's supposed to, and it will make fluorescent virus particles. And that is because the GFP is tagged to the viral protein, so it gets packaged inside of the viral particles. You can collect them the same way, and this time you make viral particles that fluoresce green. You can then take these particles and use them to infect cells that you're culturing in a separate dish, and you can image them with confocal microscopy. And this way you can actually visualize these green dots entering your cells. So this is the first experiment we did to assess what our drugs that inhibit the authentic virus were specifically doing. So we did this whole assay, but we pre-treated our target cells with each of our drugs and some positive controls. And then we imaged them and looked at where the virus was going. So on the top are the raw images that you get out of the microscope. You can see that when you add no VLP, so no virus particle, you just have these yellow cells, but there's no green. As soon as you load VLPs on to cells that have no drug, so there should be nothing inhibited here, you start to see these green puncta or green dots inside of these yellow cells. And that means those green dots are the virus. So they have successfully entered the cell here. When we treat with a drug, which is called NH4Cl or ammonium chloride, you see that there are way less green dots inside of each of these cells. There are still a few, but when we did the significance or the statistics, we found that there were significantly less viral particles per cell, which is great because this drug is known to inhibit Ebola virus entry. So then when we treated with our drug Diphylline, we saw a lot of green virus still inside of the cells. So this was surprising to us because we originally hypothesized that these drugs would inhibit viral entry, but it seems like maybe we're inhibiting a later step in the virus because the viral particles still seem to enter the cells. We also checked whether our most potent synthetic derivative, compound 1.5, would have an effect on the GFP VLP entry into cells. And you can still see a lot of green inside of these yellow cells in these images, indicating that Diphylline and our synthetic derivatives still allow the virus to enter the early endocytic system of the cell. So this means that even though our drugs work against the virus, the authentic virus, they don't work against the specific step of viral uptake. So they have to be doing something else. So the virus is still able to get into these vesicles and be packaged, But our next question was whether or not, once packaged, would they traffic deep into the cell, into these vesicles called lysosomes. So lysosomes are specific vesicles found deep inside the cell, and you can find Ebola virus in them when there is no drug treatment. So we did a similar experiment where we took fluorescent VLPs and we put them on cells, but this time we stained for LAMP1, which is a lysosomal marker. So in these images, where you see red is where a lysosome is located, and everywhere where you see green or yellow is where our VLPs are. So we wanted to know how closely related the VLP would be to the lysosome. So we basically measured the distance between the green and the red and quantified that here below. So you can see when there's no VLP added, we don't have any green, we just have our nice green stains of our cells or of our lysosomes. And then when we treated with the VLPs, but with no drug to inhibit anything, you can see that our green dots appear more yellow, and that is because they are co-localized or in the same spot as the red marker. And that means that our red and our green are really close, as they're supposed to be. When we treated with these drugs called EPA and ammonium chloride, you can see that the green dots are further away from the red dots, and that means that the drugs inhibited trafficking into these compartments. Our compound Diphylline had a moderate effect on the localization of our VLPs. So the red and the green were kind of far away from each other in these images. But when we treated with all of our synthetic derivatives, including the most potent one, 1.5, we saw that the green and the red were actually pretty close together. So our major takeaways from this are that the drug still isn't inhibiting trafficking into these deep stage vesicles. It must be doing something else against the virus. So we've already shown that in the presence of the drug, the virus can still get into the cell, it can still be packaged into these vesicles, and it will still traffic deep inside the cell. But that next step that happens in the authentic viral infection is a fusion event, and that will release the viral genome into the cytoplasm. So we use a special VLP system called beta-lactamase VLPs to model this fusion step. And so that is the next experiment we did. So we used our special beta-lactamase VLPs, which are green when there's no infection, and they turn blue when they successfully fuse and enter the cytoplasm. So you can see in a mock infection where there's no beta-lactamase VLPs added, all of our cells are green and none of our cells are blue. But when we treat with those beta-lactamase VLPs and our vehicle, DMSO, DMSO is what all of our drugs are dissolved in, so it's basically giving the cells nothing. You can see a lot of green cells, but now we have an abundance of infected blue cells. When we treat with drugs called E64D and bafillamycin, which are known to inhibit Ebola virus fusion, we do still see some infected cells, but you can see that there are way less blue cells when compared to the DMSO, which means that we've actually inhibited viral fusion. Now, when we treat with our drugs, Diphylline, or that really potent synthetic derivative, 1.5, we also see some infection, but way less than we do with no drug. And that means that our derivatives are specifically inhibiting the viral fusion step of Ebola virus. So with these few experiments, what we were able to show is that in the presence of our drugs, a cell is still able to take up the Ebola virus. It'll still package it into these vesicles, and it will still traffic deep inside of the cell. However, during the escape step, when the virus tries to escape from the lysosome, this is where Diphylline and the derivatives are exerting their antiviral effect against the Ebola virus. So I wanted to do the same thing or the same studies for COVID because we hypothesized that these drugs would be active against the virus that causes COVID as well. But we needed VLPs to be able to study COVID. And that's because COVID requires a BSL-3 lab, and we only have a BSL-2. So in this next part of the talk, I'm going to talk about a paper I published, which is where we developed VLPs to study SARS CoV-2, which is the causative agent or the virus that causes COVID-19. But first, I'll talk a little bit of background on the virus. These are the world statistics from the last week. And as of 4/8, which is just about a week ago, we had almost 500 million confirmed cases of COVID-19 around the world, with about 6.17 million deaths caused by COVID. And these are only the reported numbers, so there are likely far more. Now, we do have some prophylactic vaccines, which can prevent you from dying from COVID-19. And about 60% of the world population is fully vaccinated with a double dose of a COVID-19 shot. So that's actually a pretty good statistic, but I think we can do better. And we also need drugs to be able to treat people who still get sick from the virus. And so that is where my lab comes in. We're interested in creating these drugs. And one of those drugs we were interested in was Diphylon. So this is what the coronavirus looks like. It is a spherical particle. It has these giant spike proteins that stick out, and those are the red things. This is also what the vaccines target because it's on the outside of the viral particle and your antibodies can detect it. We also have 3 other structural proteins, which are the membrane glycoprotein, which is in blue, the nucleocapsid protein N, which is in yellow, and the envelope protein, which is in green. So these 4 structural proteins come together in cells and form this virus particle. So what happens when the virus particle comes in contact with a cell? Well, it will interact with the plasma membrane. I'm also showing a cell in the same capacity here. It will be trafficked into early endosomes, which are these endocytic compartments near the cell surface. And then they'll be trafficked deep into the endocytic system and deep into the cell into lysosomes, which I also mentioned Ebola virus enters through. So both Ebola virus and the SARS CoV-2 coronavirus fuse with lysosomes to release their genetic material into the cytoplasm. And this is also where a coronavirus infection is established. So once inside of the cytoplasm, this genome can give rise to viral proteins and also copy itself. And all of these components will assemble on these internal cellular membranes. Then they will bud into a virus particle and be released from the cell. So we sent our Diphylline derivatives back to our collaborators at Boston University who have a fancy BSL-4 lab, and they tested these drugs against the authentic coronavirus for us. So we're looking at the same figure or a similar figure as I showed at the beginning of the talk, where we have 100% virus living up here. But as you increase drug concentration going to the right, you see that the virus starts to die off. You can even see that over here at this low drug concentration for this drug, the virus doesn't even live past about 20%, even at the lowest drug concentration. That is because this synthetic derivative is extremely, extremely potent and actually one of the best inhibitors of both Ebola virus and SARS CoV-2 that our BSL-4 collaborators have seen. So we're extremely excited about this drug and being able to push it into animal studies and eventually into the clinic. So at this point, I wanted to be able to study what Diphylline does against SARS CoV-2 using VLPs, but I needed to develop a VLP system in order to do this. So again, a VLP is a virus-like particle. It's supposed to model the authentic live virus particle, but it doesn't contain a genome, so it's just kind of hollow inside. But you can see that it's still made out of the same 4 structural proteins. But they lack the proteins, which do all of the replication. So if these virus particles infect a cell, they just kind of fall apart and die because there's no genome and no replication machinery. So going back to our virus life cycle, our VLPs can model all of the steps of the virus life cycle, except for these ones that involve the genome. So we can still take those 4 structural proteins and express them in cells. They will self-assemble at these membranes and bud into virus particles that will be released from the cell. And then you can collect those viral particles and use them to infect cells and model their viral entry. So I used that same method I showed earlier, which was called transfection, where I made DNA that encoded each of my proteins, and then I gave them to the cells. The cells took up that DNA and they started to make the virus. And then I collected that virus and looked at it with electron microscopy. You can see that if you use all of the structural proteins without the spike, you just start to get these bald spherical VLPs. But as soon as you add the spike into the transfection, you start to see the spike protein stick out from the edges of the coronavirus. And that gives its characteristic crown or corona, which is where coronaviruses get their name from. We thought our VLPs looked a lot like the authentic virus as imaged by the CDC. So we were really happy with the structural integrity and morphology of our VLPs. They look just like the real virus, but they're hollow inside, so they're not dangerous. Then we used a slightly different type of microscopy to examine the outside of the cells which produced our virus. And you can see in the bottom left corner of this image down here is all of these spherical particles, which are the virus being released from the cell. And in this condition, you can see an abundance of these viral particles being released from the cell. So we thought this was really cool, but I thought it'd be even cooler if we cut open these cells and looked at the assembly of these viral particles from the inside. So we did this by using a special tag that forms a dark precipitate that you can visualize with electron microscopy. So on your far left is a picture of an entire cell, and everywhere where you see dark stain is where the virus is located. So when you zoom in on these membranes where the coronaviruses are known to assemble and bud, you can start to see these dark spheres inside of these membranes. And we believe that those are coronavirus particles, or VLPs, that have assembled in the membranes and will eventually be released from the cell. Down here in E, you can see about 6 viral particles inside a vesicle that will eventually be released from the cell into the media. So we thought this was super cool. We knew that we could make our viral particles and we could collect them, but I wanted to know if I could make them glow under the microscope. So what I did was I took our VLP and I added a green fluorescent protein tag, and this causes our virus particles to fluoresce. So if you take these particles and you use them to infect cells, you can then stain them with a red plasma membrane stain, which you see here surrounding the cell. And you see a big nucleus, which is in blue in the cell as well. But in the mock infection, where there's no virus particles, you don't see any green. But as soon as you infect with our green virus particles, you can start to see these bright green dots inside of the cell, and those are the virus infecting the cell. So at this point, I did a lot of work developing these virus particles and making them fluoresce and making sure that we could validate everything that we were doing and that it correlates well with the BSL-4 studies. So I never actually got to testing the drugs with my VLPs. So that is the next step in this project that the next graduate student will be doing. So our conclusions from this talk are that Diphylline and our synthetic derivatives specifically inhibit viral fusion of Ebola virus. And we hypothesize that they also inhibit specifically viral fusion of SARS CoV-2. We also show that SARS CoV-2, the virus that causes COVID-19, we can model its assembly, budding, and entry with our virus-like particles, which means that we can safely model these viruses inside of a BSL-2 lab, which takes a huge workload off of our BSL-4 scientists. So I'd just like to acknowledge and thank everybody in my lab that has contributed to the research that I showed you today, including my thesis committee, everybody in the Stalin lab. I'd just like to highlight our PI, Dr. Rob Stalin, and our lab manager, Dr. Nathan Dissinger, who takes care of all of our cells. The Davison lab are the people that did all of the synthesis of Diphylline and our synthetic derivatives. And then the Davy lab is that BSL-4 lab up in Boston that did all of our live virus testing for us. I'd also like to acknowledge our funding, and specifically this BOLD grant, which is the T32 that funded my research here at Purdue. So thank you. With that, I can take any questions.
Fantastic, Caroline. So good. So exciting. And I have to admit, I love your group, the group that you work with. I think you guys are dynamic and fun, and I really, I work well with your prof, Rob Stalin, and we have a good time together. But tell me a little bit, like what motivated you to get into this world of, well, research and viral research, especially with such dangerous viruses?
Yeah, I have kind of a funny story. When I first started doing research, which I actually was in high school, I got into a really cool internship program when I was 16 at the National Cancer Institute, and that kind of opened my mind to research. I had like never really thought about it before. So I got to learn a little bit about cancer research through that internship. And then my dad had cancer, and I was like, oh my gosh, I really want to do cancer research and like really make a difference in that field. So I did cancer research throughout college. But then when I applied to grad school, I came to Purdue again, thinking I'm going to do cancer research. And then I met Rob Stalen, the guy that I work for. And he was just showing me all this like amazing Ebola stuff that I couldn't believe that you could just do in a normal lab. And I was just like, wow, I really need to do that. Like that is just calling my name. And so I really knew absolutely nothing about like viruses. I took one intro virology class in college. So that was like all I knew. And I was like, yeah, I guess I'll do a PhD in viruses. Sounds great. Um, and I just like absolutely have loved it. I really realized that my interests are just in the molecular biology of disease. So I could be happy doing cancer or viruses, but I also felt like with the underprivilegedness of the people that live in Africa and are most affected by these viruses, that I was like making a difference in that part of the world by doing this type of research where they can't even do this type of research in some places in Africa because they just don't even have the BSL-2 facilities. So anything we can do can help them out. And I just feel like it like fills my heart to be able to do this type of research.
That's great, and so much passion that you portray when you're talking about it. I think it comes through, and it shows that you really care about what you're doing. Um, Caroline, you've been throwing this term BSL, BSL, BSL-2, BSL-3. I just want to make sure everybody knows that this stands for biosafety level. And the biosafety level, as Caroline was showing, there's these different levels all the way from 1 to 4. And it's the danger level of working with these pathogens. Caroline, what was the best advice that you got as you think you went through that whole cancer research period? Or maybe what is the best advice that you've gotten during your PhD as you've moved through this process?
I personally, I don't know if this will be applicable to everybody, but I'm sure it will be applicable to some people. The best piece of advice I ever got was to just slow down and think about what you're doing and like really like stop and like have a written out plan of like what you're going to execute when you go into the lab. And that way you never end up in a position where you don't know what you're doing or like you have live virus out and like you're like, oh no, I didn't do math correctly. And now I have to do something with this virus. So it's like not putting yourself in situations that are dangerous in the research.
Well, that's
But
great.
I think that was the best for me. Yeah.
That's great advice. I think that's also good advice because it helps people plan their day better, and so they feel more accomplished as they look back and everything that they had planned out, and maybe they didn't do it all, but at least some of it you've done, right?
Yeah.
Uh, That's really good. And tell me a little bit about what it was like for you as you got started. Did you have an undergraduate experience where you volunteered first in a lab to see what it was like, and then you kind of decided, yeah, this is for me, or how was it?
Yeah. I got to do undergraduate research in a computational chemistry lab at my undergraduate institution. So that was interesting because it was very different from what I do now, just sitting at the computer simulating, proteins moving in water, which I thought was cool. But I, during the summers, volunteered in like a more pharmacology lab that was hands-on. And I really wanted to be able to do something that combined both of those worlds. So now all of our Diphylon stuff was predicted from molecular docking, which I didn't talk about. But basically, we had scientists that are really good at computers tell us, like, we think that this drug will work against the virus based on our calculations. And so I was like, that's something I really want to work on. So then I came into the lab and picked it up. Um, I can't remember what your question was at this point. I don't know if I answered it.
No, I think you nailed it in terms of, what is it what is it that is really moving you towards this direction? And was there an experience in your undergrad that you had to either volunteer to get into a lab, or that you had to do something to start your career in research, or at least in your undergrad? um, and tell me a little bit, this molecular docking term, which I'm sure that the people listening, especially because they understand video games or they've grown up with video games and computers. Tell us a little bit more about this molecular docking. So what I understand that they are computational scientists. How do they do this stuff? Do you know a little bit about that?
Yeah, I'm trying to think of the best way to explain it. Um, it's like kind of like a computer game in that things are moving around and you can like, it's like The Sims where you're building a house. You can like put, your chairs down in the kitchen and a table. You can kind of set up a house per se, and then you have to add doors. But if you take away the doors, the Sims don't know what they're doing. And so they kind of do this thing with molecular docking where they like add a drug and see if it takes away any of the doors, and then they simulate to see what the people will do, but with viruses and with proteins, not with people. So they kind of run this little simulation, see what the beginning and the end points are, and that can give insight into whether a drug might work against the virus in humans. And then those people that did all the calculations can talk to people like my group who can actually do the experiments in the lab, and we can tell them whether or not their simulations are good or bad. Then they can go back and refine the math and get better every
Right.
time.
That is so important to understand, that there is a balance there, that you're working with a team of people that are each doing their own thing, and that all that information is then coming together. Um, these simulations, though, they're essentially just understanding interactions, whereas your tool really lets us see if a drug stops the virus from coming into the cell or not. And I think that's very important to realize, that just because a drug has a close interaction with a protein, does that mean that it's always going to stop that virus from coming into the cell?
Yeah, that's a great question. You would think yes, but the answer is truly no. Lots of times the computer says, hey, this is going to work and it's going to work great. And then you get into the lab and it doesn't do anything. And you're like, oh no, what could have possibly gone wrong? And it's because there's just more to life than our simulations can currently capture. There's more to life than just force equals mass times acceleration, which those simulations are based off of. And there's more to life than just dipole interactions. So, and that's something a cell model like in our plates and our VLPs can model, but that we can't quite get to with just a computer yet. So that's why experimental sciences are still going to be around for a long time.
Really, really neat. And also, I guess this whole field of machine learning and artificial intelligence, how it's going to excel and help us create better designs, better models, better drugs at the end of the day, right?
Mhm.
I mean, that is such a fascinating field. So data science is a field that is exploding. I can imagine you've seen that as well. All the data that you have collected and how you've assembled that data. I wonder how much data that you have not assembled that you've collected as well, right?
Yeah, it's on the order of like hundreds of gigabytes of data. It's a lot. It's kind of funny when you look back at like what somebody did in 1950 to get a PhD. It might be like a couple gigabytes worth of data, which was a lot at the time, right? And now, like, it's so routine to just like get a couple terabytes. It's really crazy.
Unbelievable. That's phenomenal to hear. Caroline, how do we handle all of this stuff? I mean, what is the advice that you would give to somebody who wants to get started, somebody that wants to learn, what's happening in the lab beyond what they've learned in the classroom?
Yeah, my best advice is to like get some hands-on experience. The best way to do that is to either apply for internships through like the National Institutes of Health or the National Cancer Institute, or every undergraduate university. Most of them have internship programs for the summer. And if you don't want to fill out a formal application, or you don't get offered a position through the formal application, just Google search research at Purdue University, find a professor that does something that you're interested in, and send them an email and say that you're interested and you would love to either like work for them or just come check out the lab and see what's going on. And most of the PIs, are really nice people, and they will like love to have you come and see what they're doing. Especially like, I don't think there's an age that's too young. I mean, I was 16 the first time I was allowed into a lab, and I think it was a great experience. I didn't totally understand everything I did at 16, but I learned so much in a lab that they don't teach you until grad school in a class. So I just, yeah, definitely go find some hands-on experience in a research lab and see if you like that type of work.
That's great advice. And what about, when you're in your undergrad, don't they give you like a lot of homework and you have to do a lot of classes? How was it juggling all of that with getting this experience in the lab? Were you able to handle it okay?
I didn't do like research during the semester, my first couple of semesters. I only did it in the summer for the first year when I was like very class heavy. But then my sophomore year, I started to like add in, like 5 hours a week of research. And then I slowly pulled that up to about 20 my senior year. And that's because I wanted to be doing that much. I think once you are in that invested, it becomes really easy and you just want to be there. But I very slowly added credit hours until I was almost my last semester doing mostly research and like just a couple classes. So that's kind of how I did it. I know other people that did start doing research their first couple of semesters, and I think they really loved it. So like if you have time in your schedule and you can like really, if you feel like you have time to like really go put that amount of brain energy into it, like go ahead and do it. Otherwise, like it's totally okay to wait until your sophomore year as well.
That's great. And did you think you would feel more or less courageous had you peeked in before, or even just had a look or conversation with a prof ahead of time, so that you can ease yourself into that position?
Yeah, definitely. I just talking to people and asking like, hey, what is this going to be like is like a huge benefit, because they can at least tell you like this is what you can expect. And you don't come in totally blind to what's about to happen. Not that it should be scary, but it can be a little overwhelming at first. You walk into a lab and you've never seen most of the things in front of you. It's a little weird. But just find a good PI that can walk you through everything. And that's, the best advice I have.
I agree. And if it's not a good PI, even just a good student or a postdoc or
Good
somebody in the
mentor.
lab,
Yeah.
yeah, that can just take you around, show you, and explain a little bit about what's happening. It might, as you said, it might be all over your head. You might need to hear it a few times before it sinks in. That's great. I want to open it up. I don't know if Mr. Rackless or anybody else online wanted to ask questions. And I know I'm hogging the mic here. I was just so excited to get to talk to you, Caroline, and ask some of these questions. I have more questions if nobody else is going to ask any. And so I'll take it. Caroline, um, you mentioned the impact that you're having in doing this work. It must be something that doesn't necessarily motivate you every day to wake up and go do this, but it probably does overall give you that kind of ambition to keep going. What's been your favorite part about, doing your PhD or this experience at university, the different universities that you've attended?
I have like two answers to that. My first one is like I loved all the people that I met during my PhD. I've learned so much just from talking to other scientists and other people in, this world. And my second answer is I loved doing the COVID research. I just thought it was so cool. And I couldn't believe that like a virus pandemic broke out while I was doing my PhD in viruses. And then I was like, not only did that happen, I got to study it. And like we got to do so much fun stuff in the lab that was like really time sensitive because we're like, we want to be the first ones to publish. Like we wanted to like make the like first paper and we did, and it was super fun. And I just that experience was unlike any other. And I feel like I could have only ever gotten that experience at Purdue. So I'm pretty happy with my graduate school experience here.
Well, congratulations. That's quite a feat. And I can imagine the excitement in the lab and trying to get all that stuff out on time. And especially because the world was waiting for some of these answers that you guys were finding on how the virus is entering and, the different life cycle of the virus. It was great, great work. And I'm so glad to at least be close enough to it to have seen some of that working your continued work. So congratulations on all of that. um, I wanted to ask you, from all the people you admire, what is a common trait about them that you admire, and what is it about that you might want to adopt in your life, in your career?
They like to question everything. That's one thing I found in a lot of people that I admire. And like not in a bad way, but in a like, I don't understand what's in front of me, so I need to ask some more questions to figure it out in terms of the experiments. And like we get data back sometimes where you expect like black or white and it turns out gray, and you're like, what do I do with that? I don't know what that means. And so being able to like take gray information and design a black and white experiment from that is something that they all have in common. And also they're all very friendly. Many of the like best scientists I know are actually like real people, and they're like very nice. And there's just no need for like the stereotypical professor mindset. I feel like the best science comes from collaborations and working together, and almost everything I've done has been collaborative. And it just has been a really great experience.
Fantastic. That was a great answer and a great way to end it. I want to thank you, Dr. Caroline Plescia from the Department of Medicinal Chemistry and Molecular Pharmacology here at Purdue University. Again, my name is Tommy Soares. I welcome you to keep joining us for these lecture hall series where we speak to wonderful scientists like Caroline, and we get to hear a little bit about their experience. Thank you, everybody, for joining us again, and we'll see you soon.
Thank you.
Thank you so much.